Welcome to the Alberta Forage Industry Network Sustainable Forages webinar and podcast series. You might be asking, who is Alberta Forage Industry Network? The forage industry is significant in Alberta. According to the most recent census, there are over 28.8 million acres of forage land in Alberta. Up until 2002, the Alberta Forage Council represented the interests of the forage industry in Alberta. However, in 2002, the council merged with Applied Research Associations to become the Agriculture Research and Extension Council of Alberta. ARICA has evolved to become a broader umbrella organization representing the whole agriculture industry in Alberta. Several consultations were held with all facets of the Alberta forage industry between the years 2007 and 2010 to see if there was an interest or need to have an organization representing the forages in Alberta. This resulted in the formation of the Alberta Forage Industry Network Society, or AFIN, in 2010. AFIN is directed and managed by a volunteer board of directors who are located across the province. For more information and contact information about the AFIN board of directors or how you can become a member of AFIN, please refer to the Contact Us section of the AFIN website at www.albertaforages.ca.
We have about 20 people already, so let's start the webinar. I think it is just after 7. I think I'm Surya Acharya. If you don't know me, I chair the Alberta Forage Industry Network board, and that's about all I would say in terms of my introduction. I think we are here for the webinar today. And before we do anything more, I think I should acknowledge that Alberta Forage Industry Network respectfully acknowledges that we live and work on traditional lands of the Indigenous peoples, Inuit, First Nations, Métis of Canada. We honor the ancestry, heritage, and gifts of the Indigenous peoples and give thanks to them. And my job this evening is to introduce our speaker, Dr. Malinda Thilakarathna. He is an assistant professor in agronomy and plant microbiology interactions at the Department of Agricultural, Food and Nutritional Science, University of Alberta. He earned his PhD from Dalhousie University, Department of Biology, followed by postdoctoral research at Department of Plant Agriculture, University of Guelph. His current research at the University of Alberta focuses on various aspects, including biological nitrogen fixation in legume crops, below ground nitrogen transfer, soil amendments of plant and soil health improvement, and plant responses to biotic and abiotic stresses. And that is the main reason why we want to hear Malinda this evening. Malinda's research program combines both basic and applied approaches, encompassing diverse crops such as pulses, forage legumes, forage grasses, cereals, and oilseed crops. His collaborative efforts extend across different disciplines and sectors, including academia, industry, and government. Notably, the industry's keen interest in his work is evident through substantial funding exceeding 250,000 in industry cash. His research program is further supported by various funding sources, including Natural Science and Engineering Research Council of Canada, which we know as NSERC, and also results-driven agricultural research, RDAR, and MITAX, Rangeland Research Institute, and industry, Prairie Mines and Royalty, ULC, and Huma Terra. I don't even know some of these things. I never got funding from all these all these different funding agencies in my lifetime. But anyway, Malinda is doing very well with all the funding. With he has many peer-reviewed publications and numerous presentations at national and international conferences. He has established himself as a prominent researcher in the field. In recognition of his outstanding contributions, Malinda received the Canadian Society of Agronomy Early Career Agronomist Award in 2021. In addition to that, he was honored with the Faculty of Agricultural Life and Environmental Science Research and Innovation Award in 2023. Without further ado, I would let Malinda give the seminar this afternoon. Its effect of drought stress on plant growth, nitrogen fixation, soil nitrogen availability, and soil microbial diversity in forage legumes. Malinda.
All right. Thank you very much, Surya, for the nice introduction, and also thanks for inviting me to give a presentation for this webinar series. And I'm excited to talk about this project. What we have done so far at the University of Alberta. So basically, I started my research program, just before COVID. So I joined University of Alberta 2019 November. So yeah, just before COVID time. So today I'm going to talk about the effect of drought stress on plant growth, nitrogen fixation, soil nitrogen availability, and soil microbial diversity in forage legumes. I think most of you are well familiar with what is biological nitrogen fixation or symbiotic nitrogen fixation in legumes. Legumes include pulses and also forage legume. So what happened here, this is just to give you some brief introduction. So we know legume plants, they produce root nodules. So in the root system, so what happened here, inside the root nodules, so these are alfalfa plants and these are root nodules. And if you take a cross section, so there are special structures, and inside these root nodules, there are millions of nitrogen fixing bacteria called rhizobia live inside. So what they basically do, they can take atmospheric nitrogen, which is not available for plants because plants, they don't have an enzyme called nitrogenase in order to make this nitrogen gas into the usable form like ammonium. So what happened, these bacteria can convert this atmospheric nitrogen into ammonium and supply it to the plants. Okay, and there's a symbiotic relationship between the rhizobia bacteria inside the root nodules and the legume plants. So as you can see the picture on the right side, so these bacteria, they fix nitrogen and supply to the plant, and as a return, plants provide sugar, like the photosynthesis they're produced by the plants as the return. So there's a clear symbiotic relationship between this nitrogen fixing rhizobia and the legume plants. So although it looks like very simple process, plants produce root nodules and they fix nitrogen and supply to the plants, that process is very complex. So here, I just want to like highlight here, some of the key pathways, that needs to go through in order to produce successful nodulation and nitrogen fixation, just to give some idea about, how complex this process is. So in any legume root system, they have these tiny root hairs, and these root hairs, they release a specific chemical compound called like flavonoids. So this flavonoid, it acts as a like kind of like, signaling compound, like it sends message to the rhizobia in the soil. So once these chemical compound received by the soil rhizobia or rhizobia that we introduce as inoculant, there's some, there like regulation of some of the genes inside these rhizobia. It's like, rhizobia get alert by these plant compound, flavonoid compounds. Then this rhizobia release a specific chemical compound called nod factors. And these nod factors, that chemical compound will be identified by the plant. So once plants identify this chemical compound, so the bacteria can attract towards these root hairs. Then what happens? These bacteria can be trapped by these root hairs, and this special process is called like root hair curling. So once root hair curling happens, these bacteria trap here, then plants form a special structure called infection thread, and through this infection thread, these bacteria can direct towards the inside of the root system. So that's where the nodulation process start. Then once plants form nodules, so inside these bacteria, I mean nodules, there are many bacteria, they can convert this atmospheric nitrogen into usable form like ammonium with the help of the enzyme called nitrogenase enzyme. So this is a complex process, like we see, although we see, the root nodules, then we think, if they are red color, right, they fix atmospheric nitrogen, they supply to the plants. But this, just to give you an idea, like this is like very complex process. Okay, so what happened after, so this bacteria fix nitrogen? So as you can see, this picture here on the right side, so nitrogen is reduced into ammonium inside the root nodules. Then this ammonium is converted into like amino acid, especially like glutamine. This amino acid can be transferred to above ground plant parts, including like leaves, especially like in forage legume. The leaves are the most important part. Then this nitrogen compound can be, help to like develop chlorophyll and produce nitrogen-rich forage for, animals. Like when it comes to like seeds, especially like pulses and also oilseed crops like soybean, and this nitrogen can also help to increase the nitrogen content. Okay, so just want to give, some idea about, like drought stress. Here you can see these are like, map Canada, and these are like some models prepared by the Natural Resource Canada, and this shows like, what's going to happen in the future in terms of drought stress. And this bar indicates, blue color is, kind of like wet, and when it go to like, dark color, including yellow, then bright red color, it shows the, drought areas. So as you can see here, this is 2011 to like 2014, and there's another map predicting like what's going to happen between 2041 to like 2017, then like 2071 to like 2100. So as you can clearly see, this dark brown area, it's kind of like expanding, especially in prairies. So you can see, in Alberta, so we can see clearly the drought is going to be like increasing. There's a like increase in trend. So we can, in the future, we can, mostly experience like more drought situation. I think we are already experiencing this drought condition. I think like 2 years ago, like we had a really bad drought condition, and even like this winter, right? The precipitation, like the amount of snow we received, like it's relatively low compared to what we used to get. So I'm pretty sure, in this spring, most of the producers is going to have like, hard time. So there'll be not enough snow coverage, so that can create some moisture issues and maybe early drought stress during spring, unless we get, more snow or more rainfall during springtime. So drought, so it's going to be like, really problem we already experienced, and I think, it's going to be a like big problem in the future as well. Okay, so in our lab, so we did like some experiment to test, how this drought condition is going to affect like forage legumes. So we conducted some greenhouse trials. So basic concept here is, like you can see this legume plant. So when there's drought stress, that can affect plant growth, photosynthesis, leaf chlorophyll, that can really affect the shoot growth and nutritional content. So what's happening below ground? This drought condition, can create low moisture environment, and that can affect directly nodulation as well as like root growth, and that also going to affect the nitrogen, biological nitrogen fixation. And since the low moisture availability also going to affect root growth, that can affect like release like rhizodeposit, that means like release of different compound by the root system, includes some carbon compound, nitrogen compound. So that's going to affect like soil nitrogen availability. And some of these compounds, especially like carbon and nitrogen compound, they're very important in like soil microbes, and also some enzymes, which can decompose and, release different nutrient. So everything, linked together. So we think, this drought condition can, have like big impact, like both above ground as well as below ground as well. So based on this hypothesis, we designed some experiment. So in the first experiment, I'm going to talk about here the research objectives we had to evaluate the effect of different drought stress on forage legume growth, nodulation, nitrogen fixation, soil nitrogen availability, soil enzyme activities, that's important for nutrient cycling, and also like soil microbial diversity. So what we did here in this experiment, so we pre-germinated seeds. So here we were using both red clover and alfalfa. Then so we planted seeds into like, this is like soil-based mixture. So we collected soil sample from the field, then we brought them here, then we mixed with a little bit of sand because we wanted to evaluate the nodulation profile. Then we inoculated plants with alfalfa. We inoculated with specific rhizobia called Rhizobium millileuti, and red clover we inoculated the Rhizobium leguminosarum biovariety trifoliate. Then after 2 weeks of plant growth, we basically like labeled, or like we supplied, this is like N15, that's the isotope-based nitrate mixture. We applied very low concentration. So this is to label plants. Then we can measure nitrogen fixation using a specific method. So that's called isotope dilution method. So that's the main purpose why we labeled these plants with this isotope, nitrogen isotope. Then we grew the plants. Then when they reach to like early flowering stage, we induce 3 different moisture treatment. So these are the 3 treatment we applied. The 80% field capacity, so that's a well water treatment. Then 40% field capacity, so we consider as a moderate drought. Then we also had another treatment, 20% field capacity moisture. So that's the severe drought condition. So after we induce drought treatment, okay, so we left the plant 3 weeks. Then after 3 weeks, we measured different plant physiological parameters. So we measured leaf chlorophyll content. We measured plant like leaf photosynthesis using a multi-spec tool. So as you can see here, this instrument is called multi-spec tool. Then at the same time, after 3 weeks, we installed these PRS probes. So these are called like plant root simulator probes. So these probes actually, it's available from one of the Canadian company in Saskatchewan. So Western Ag Company. Maybe some of you familiar with this PRS probe. So what these probes does, they do actually, there's like anion probes and cation forms. So once we buried them inside the soil, so there's a membrane, and this membrane absorb different anions and cations like nitrate, ammonium, sulfate, like different anion and cation in the soil, and it kind of like become equilibrium. So whatever the ions absorb in this membrane, or the nutrient absorbed in membrane, actually it represents the available nutrient in the soil. And we are mostly like specifically like important like concerned about nitrogen availability, especially nitrate and ammonium. Then at this stage, after 3 weeks of drought, we had to add water, because if it is dry, the pH probes they won't be able to absorb any nutrient. Then we won't be able to measure any anions or cations. So we supplied moisture back to the 80% capacity. Then we left 1 week. Then we remove the pH probe and use to analyze soil nitrate and ammonium availability. So after 1 week, we harvested everything. Then we collected shoot and root biomass. Then we wash all the roots. Then we counted a number of nodules, and we also measured the nodule weight, nodule dry weight. Then we measured nitrogen fixation, as I mentioned earlier, using that isotope dilution method. Okay. Then at the same time, when we did sampling, so we collected soil samples representing rhizosphere, especially like rhizosphere means like the soil immediately attached to the root surface. Then we analyze soil microbiome or soil microbe presence in the soil. And we also collected soil samples like rhizosphere soil samples. Then we analyzed them for soil extracellular enzyme activity. Okay, so here are the results. So the first row, it represents alfalfa, and the second row, it represents the red clover. As you can see here, this is 80% field capacity. Plants are, growing pretty good, healthy. And this is like moderate drought, 40% field capacity. You can see some reduction in growth. And when it comes to 20% field capacity, severe drought, and you can clearly see there's like significant reduction in plant growth as well. Okay, and this figure shows the leaf photosynthesis, and I'm going to explain what's on the x-axis and what's on the y-axis. So every figure I am going to talk about from here onward. So we have listed the first graph is alfalfa, the second graph represents red clover, and there are 3 different moisture treatment. The 20% field capacity, that's the severe drought. Then 40% field capacity, that's the moderate drought, and 80% field capacity, that's a control, 80% well-watered condition. And here y-axis it represents the photosynthesis. So interestingly, we didn't see any significant differences between different soil moisture conditions. So what we were expecting actually is we thought like 20% for the severe drought actually really reduced the leaf photosynthesis. So in alfalfa, we didn't see any differences. So what we think what happened, maybe like once we expose plants to like prolonged period like 3 weeks, so maybe plants kind of like get used to the this moisture deficit condition, and somehow they able to manage the photosynthesis. But I think if we measured photosynthesis maybe readily after we induced drought, probably we could have seen reduction in photosynthesis. So same thing with red clover. We didn't see, like statistically, there were no significant differences, but you can clearly see when it's come to 20% and 40% field capacity, like drought treatment, we can actually see numerically low value compared to the well-watered treatment. Then we measured leaf chlorophyll content. Again, same treatment on the x-axis. And in alfalfa, actually, what we found, there were no differences between the control well-watered plant and the moderate drought, but we found like reduction in leaf chlorophyll under severe drought stress in alfalfa. But opposite trend, kind of like opposite trend found with red clover. So what we notice, so when it comes to severe drought condition, the leaf chlorophyll content was much higher compared to the control well-watered treatment in red clover. So it looks like, under red clover, there's like nitrogen accumulation happen here. Okay, so this figure shows the plant biomass, which is important for producers. So this is a shoot dry weight. And the alfalfa, you can clearly see compared to the control or the well-water treatment, the drought stress drastically reduced, even under moderate drought stress and also like severe drought stress, significantly reduce the shoot biomass. So same thing with red clover as well. So you can see the biomass significantly reduced under moderate drought and severe drought. Then we also measure the root dry weight. And again, here you can see in alfalfa compared to the well-watered control. So there was a significant reduction in root dry weight in severe drought treatment. And when it comes to red clover, under both moderate drought conditions and the severe drought condition, the root biomass significantly reduced compared to the well-watered treatment. Then we counted the number of nodules because nodules are important for nitrogen fixation. And here alfalfa, you can clearly see both in red clover and alfalfa, especially alfalfa nodule numbers significantly reduced. This is the total number of nodules per plant, significantly reduced under moderate and severe drought state condition compared to the well-watered control. Same trend with red clover, but there was no significant differences between moderate and well-watered condition. But when it's come to severe drought condition, the number of nodules drastically reduced compared to the well-watered plants. Then we measured the nodule dry weight. So interestingly, we didn't find the nodule dry weight. They were not significantly different between the moisture treatment in alfalfa. So what we found when it comes to like moderate and severe drought conditions, so actually plants tend to have like large nodules under drought stress. So maybe that's a like plant mechanism, they are trying to maintain like maybe less number of nodules, but having large nodules somehow, support the nitrogen fixation. But we didn't see that trend in red clover. So there was a reduction in nodule dry weight, especially under severe drought condition. But there was a reduction in nodule dry weight when it's come to moderate drought condition compared to the well-watered, although it was not significantly different. So basically, like drought stress reduced the number of nodules and also nodule biomass. Okay, then we calculated the symbiotic nitrogen fixation. So as you can see here, the y-axis, it shows like percentage NDFA. So NDFA stands for nitrogen derived from atmosphere. So what's that mean? So that means like out of 100% of plant like shoot total nitrogen. So here it's like, 70% of the nitrogen came from biological nitrogen fixation. So in alfalfa, we didn't see any significant differences between the treatments. So that means like drought didn't have significant effect on reducing nitrogen fixation. But still, you can see there's like clear reduction trends, although they were not significantly different. I think because we had some kind of like outliers here. So maybe I still believe like drought like reduce the biological nitrogen fixation. But on the other hand, red clover actually clearly shows like here even like close to 20%. So only like 20% of the nitrogen came from biological nitrogen fixation when it's come to severe drought stress compared to the well-watered plant. So red clover, I believe it's more sensitive to drought stress when it's come to nitrogen fixation compared to the alfalfa. So based on this value, like percentage nitrogen derived from atmosphere, we calculated the shoot total nitrogen fix. So this shows like milligram of nitrogen per plant. So you can clearly see the total shoot nitrogen fixation like significantly, like these values like 150 here. So this is like 50. You can clearly see like few fold of reduction in nitrogen fixation when it comes to moderate drought and severe drought stress. So same trend here, like especially under severe drought stress, the shoot nitrogen fixation was much lower in red clover compared to the 80% field capacity well water treatment. Excuse me. So this figure, it shows the shoot nitrogen concentration. So in alfalfa, you can see there was a significant reduction, like the shoot nitrogen concentration, it was very low compared to the well-watered plants, but we didn't see any differences between these well-watered and the moderate drought treatment. When it comes to red clover, again, we didn't see significant differences between these three moisture treatment in terms of like shoot nitrogen concentration. Then we calculated the total shoot nitrogen content. That is important again for producers. That like they care about how much green color, whether it's rich in protein, right? So again, you can clearly see under moderate drought and also like under severe drought condition, the shoot total nitrogen content is much lower compared to the well-watered control treatment. And here, like red clover, we didn't see any significant difference in terms of statistically, but still you can see there was a reduction in terms of like numerical value. There was a reduction in shoot nitrogen content when it's come to severe drought stress compared to the well-watered control plants. Okay, then this is very important. I feel like this is very interesting finding. So soil nitrogen availability. This figure shows the soil nitrate availability. And so what we found, like compared to the well-watered plants, like when we expose to drought condition, this is moderate drought and severe drought. So after 3 weeks of drought, so we found the soil nitrate level much higher under severe drought stress compared to the well-watered plant. So same trend with red clover, you can see here under severe drought stress, the soil nitrate levels were much higher compared to the even like moderate drought stress and also severe control, 80% field capacity control plants. Then we also calculated the soil available total nitrogen. So this is the combination of soil nitrate and ammonium. But what we found in the soil available ammonium content is much lower compared to soil nitrate. So these figures represent like both ammonium and nitrate. Again, the same trend, the soil available nitrogen, it was much higher under severe drought stress, following drought stress compared to the well-watered plants, and same trend we notice in red clover as well. So I think this is important for like forage producers. So what happened, if there's a like prolonged drought condition, so that means after drought condition, so there will be like more nitrogen available in the soil. So maybe that nitrogen, if we get like maybe severe rainfall, some of the nitrogen can be leached out from the system. But if you get, maybe like moderate rainfall, so that nitrogen can be available. So some part of the nitrogen, if it's like grass legume mixture, so grass can readily absorb this nitrogen, and also like some of the legumes also can absorb this nitrogen. Okay, the next one is soil enzyme activity. So we measured using soil samples, and this one here, it shows the enzyme called cellobiosidase activity. So that this enzyme is important for carbon cycling. So we didn't see significant differences between the moisture treatment, but as you can see, they under severe drought stress and also moderate drought stress, there was a like relatively low carbon cycling activity compared to the well water treatment. And when it's come to red clover, we clearly saw significant reduction under severe drought stress in terms of cellobiosidase activity compared to the well-watered 80% field capacity treatment. Then we also evaluated another enzyme. It's called N-acetylglucosaminidase. That is important for nitrogen cycling. What we found with alfalfa, there's moderate and severe drought stress. Under moderate and severe drought stress, these enzyme activities significantly reduced compared to the well-watered plants. But when it's come to red clover, we didn't see that trend. So maybe this soil enzyme activity might be different based on the the forage species as well. Okay, so the next one is so we evaluated like soil microbiome. So what's shown in here, like the left side picture represents the alfalfa, the right side picture represents the red clover. And we collected soil samples from bulk soil, away from the root system. And we also collect soil samples from rhizobia. So rhizobia soil means like immediate, the soil surface, soil attached to the root surface. So what we found actually, the rhizobia represent like completely different microbes. And the bulk soil, like rhizobia soil and the bulk soil, they have different microbial community when it's come to alfalfa. And red clover also, like we found, like the bulk soil and rhizobia soil, they have different microbial. But still, some microbes, are common for both bulk soil and rhizobia soil. Then after that, we dig into like more details, looking into like specific microbial, families. And we found some differences for specific microbe. So the first figure, it shows bulk soil acetobacteria. So this bacteria is important for carbon cycling, especially through the breakdown of plant biomass. So what we found, there was a reduction under moderate drought stress compared to the well-watered condition. Then the second one, there's another bacteria category called proteobacteria. This is in the bulk soil. So proteobacteria is important for carbon, nitrogen, and sulfur cycling. They have different roles. And what we found under severe drought stress, this proteobacteria abundance increase compared to the well-watered treatment, and also like moderate drought treatment. The third category, it's called Spingomonadiaceae, and this is important. These bacteria actually produce different plant growth hormones and also help to like promote root growth. So what we found under severe drought stress and also like here, like moderate drought stress, the abundance increase compared to the well-watered control. So what we think here, the plants are under stress, so maybe plants kind of like giving some signals to these microbes, they can colonize around the root system and they can secrete, some plant growth hormones and, increasing root growth because, now plants are experiencing drought stress. So plants kind of, having some mechanism to like increase root growth and maybe absorb moisture from like deep soil layers. So the last category is called Nocardiodacea. So this has a role in especially like tolerant to desiccation, like drought stress. So as you can see here, the severe drought stress, these microbial abundance increase compared to the well-watered condition. Okay, so we found different microbes like respond different ways to this moisture stress in forage legume. Okay, so based on this study, main conclusion we can take this drought stress reduce the nodulation parameters, including number of nodules and nodule dry weight. Then it significantly reduced the plant biomass, including shoot and root. Then it changed the like leaf chlorophyll content, especially in alfalfa. So we found an increase under moderate drought stress and reduce under severe drought stress. But in red clover, we found increase under moderate and both moderate and severe drought stress. And Interestingly, we didn't find any differences in terms of photosynthesis between the well-watered plants and the drought plants. And in terms of extracellular enzyme activity, in alfalfa, we found like nitrogen cycling enzyme activity was reduced. So maybe one reason is that, we found like increase in soil nitrogen availability. So that increase in nitrogen availability could be due to, plants, maybe some of the nodules can die because of moisture stress. Then some of the nitrogen compound can release through these nodules, and also some roots, they can release different nitrogen containing compound, include like different amino acid that also help to increase the nitrogen availability in the soil. And in red clover, we found carbon cycling enzyme activity, it got reduced. And yes, here like we found like drought increased soil nitrogen availability following drought stress. And in terms of microbiome, we found like shift in the relative abundance of some of the key bacterial taxa under drought stress. Okay, so that's the first study I want to present today. Then I'm going to talk about kind of like similar study we recently completed. So in this study, we thought like rather than growing just legumes, so why don't we grow both legumes and grass together, and also grass separately as monoculture, and see what's going to happen under drought stress. So the title is Effect of Drought on Red Clover Grass Mixed Stand compared to grass monoculture stands. So here then our research objective was to assess how plant growth, symbiotic nitrogen fixation, forage nitrogen production, carbon and nitrogen dynamics, and water use efficiency are impacted under different drought severity conditions and after recovery phase following drought in red clover, Timothy mixture stand compared to the grass monoculture stand. So again, we conducted a greenhouse study. So we brought soil samples from the field. Then we filled these pots. And the way we arrange plants, as you can see here, this is like, so we had 9 Timothy plants here. And for the red clover grass stand, so we established 6 grass around the border here, and we had 3 legume plants in the center. So this one shows the legume grass mixed stand. As you can see, the red clover plants and Timothy plants around, and this is the grass monoculture setup. And here we again, inoculated plants with rhizobium leguminosarum biovariety trifoli in order to induce the nodulation and nitrogen fixation. And here on the right side is a picture, representing our greenhouse study at kind of like early stages. Okay, so then how we impose our drought stress? So what we did, so we started this experiment, and we were maintaining the moisture levels in the pots under 80% field capacity. So that's well-watered plants. Then we had like legume grass mixed cultures and grass monoculture stand. Then we grew these plants for 8 weeks. So that's the initial growth phase. So after 8 weeks, so we induced drought. So basically what we did, we stopped adding water, we stopped the irrigation, then let them like dry. And half of the plants we maintain at the 40% field capacity, that's the moderate drought. Then we kept, maintaining that 40% field capacity for 4 weeks. And the other group, we kept drying until they reached to like 20% field capacity. Then we maintained this moisture level again for 4 weeks. So we had 3 treatment, 20% field capacity, like similar to the previous experiment, severe drought, 40% field capacity, moderate drought, then we had 80% field capacity. That's our control well-watered plant. So we maintained drought phase for 4 weeks. Then at the 4 weeks, we sample one set of plants, and the remaining plants, so we we supplied moisture here, adequate moisture here. So brought them back to 80% field capacity, well-watered plants. So this is the recovery phase. Then we maintain plants for another 4 weeks. So we had 3 phases: initial growth phase, drought phase, then the recovery phase. So after 4 weeks of recovery phase, again, we sampled these plants destructively. And so here are some of the pictures. So you can clearly see, and this is the panel A represent the drought phase, and the bottom panel represent the recovery phase. And the first picture here on the first panel, so on the left side, that's the mixed stand, red clover and timothy mixed culture. And this picture on the right side, that's the monoculture timothy stand. This is under 80% well-watered condition. This picture, the top panel, middle picture, it shows again left side the mixed stand, then right side monoculture stand. This is under moderate drought condition, and you can see reduction in plant growth. And this is the severe drought condition, and you can see plants kind of like collapse when it's come to severe drought stress. Then what happened during the recovery phase? So you can see control plants, looks good. And when it's come to moderate drought, they have reduced the biomass. And when it's come to severe drought state, some plants kind of like able to recover a little bit as well. Then so we measured different parameters. So this first figure, this is under the drought phase, 4 weeks of drought. Then we measured plants destructively. And on the y-axis, it shows the shoot dry weight, gram per pot. And so here, I want to explain on the x-axis, this is the control treatment. And the first column is under mixed stand, and the green color bar, it represents the grass monoculture, and yellow color bar, it represents the grass biomass under mixed culture. And this blue color bar, it represents the legume for the red clover biomass. So in the mixed culture, we have both legume biomass and as well as like grass biomass. And when it's come to monoculture grass, it's just the green bar. Okay. So similarly, we have like moderate drought and also severe drought. Okay. So like here, the pattern is different compared to the experiment that I explained earlier. So here, like left side, we have the 80% for the control treatment, then moderate, and the severe drought towards the right side. So you can clearly see the legume biomass significantly reduced under moderate and severe drought stress, these blue bars. So what happened to the grass biomass? So especially under grass mixed stand, we didn't see any differences in plant biomass, even either control, moderate, or severe drought stress condition. And same trend with grass monoculture. So we didn't see reduction in shoot dry weight, even under severe drought stress compared to the monoculture grass, even under like 80% well-watered treatment. Then this is the recovery phase. So after drought phase, we let them grow for another 4 weeks. And again, this represent the shoot dry weight. You can see, like plants gain some more weight, but still you can clearly see that the biomass during the recovery phase, especially under severe drought state, it was lower compared to the well-watered condition. But you can see there's no difference between the well-watered treatment and the moderate drought treatment. Actually, like plants were able to recover, especially like red clover plants were able to recover during the recovery phase, but still you can see there's a little bit reduction in plant shoot biomass when it's come to red clover. So how about grass biomass? So under mixed stand, we didn't see any differences between the shoot dry weight in terms of like moisture stress. And same trend with grass monoculture, the green bar, we didn't see any significant differences in terms of like shoot dry weight, although you can see, there's the mean value is a little bit higher here under well-watered condition in grass monoculture. Then again, we did like similar calculation. So here again like shoot biomass, but here we represent the gram per plant, because we had like total 9 plants in each plot, each pot. When it's come to mixed stand, we had like 3 legumes, like 3 red clover plants and 6 timothy plants. So you can clearly see drought space, the red clover biomass significantly reduced. And when it come to the grass, we didn't see any differences between the treatment, but you can see the shoot dry weight per plant. Actually, in grass under mixed stand, it was the plant biomass per plant, it was actually higher compared to the grass monoculture stand in this situation, and also with moderate drought as well as severe drought condition. Then even after recovery phase, we did the same calculation. What is the shoot dry weight per plant? And you can see these blue bars, the red clover biomass, it was significantly reduced even under moderate and severe drought stress compared to the well-watered plant. And during the recovery phase, we didn't find any differences in terms of plant biomass in grass, either growing monoculture or mixed stand. And also we didn't see any significant differences between the shoot dry weight per plant between either they grow in mixed stand or whether they grow as a grass monoculture. Okay, so this figure shows the shoot total nitrogen concentration. And yeah, of course, legume plants, they have higher shoot nitrogen concentration, as you can see here. And when it's come to like severe drought stress, the red clover had relatively low shoot nitrogen concentration compared to the moderate drought stress and also like well-watered control. Actually, there was no significant difference between the well-watered control and the severe drought stress. And also like we didn't see any differences in terms like shoot total nitrogen concentration when it's come to grass mixed stand or grass monoculture stand, as you can see here, that's orange color and green color. But you can clearly see the shoot nitrogen concentration, it was like very low compared to the legumes. Then here we calculate the shoot total nitrogen concentration at the recovery phase. And during recovery phase, you can see the shoot nitrogen concentration. Actually, it was higher under severe drought stress condition compared to the well-watered plants as well as moderate drought stress. This is like red clover. When it comes to grass, you can clearly see the green color bars, like sitting at the bottom. So that means the shoot total nitrogen concentration, actually it was lower in grass monoculture, like compared to the grass mixed stand. So maybe what happened, so we think there may be some nitrogen transfer from like red clover to these timothy plants during the recovery phase. So maybe some of the nitrogen released from the red clover plants and they were absorbed by the timothy plants. Okay. So it's a benefit, like growing these grass and clover together. And this picture, it clearly shows, this is the timothy monoculture. This is under 40%, I think, yeah, 40%. That is moderate drought condition. You can clearly see the color of this timothy. It's like pale yellow, greens color. And when it comes to like red clover timothy mixed culture, you can clearly see these grass, they're like healthy, looks happy, right, compared to the plants on the left side. Okay, then we calculated the CN ratio. That is important because we like for forages, we try to have like low CN ratio, more nitrogen, and you can clearly see, yes, of course, legumes, they have low CN ratio compared to the grass, and you can see here the orange one orange bars that represent grass under mixed stand, and they were having low CN ratio compared to the grass monoculture. So that's a good thing. Same thing with the recovery phase, and you can see even like more prominent. Yes, red clover, they're having low CN ratio. Then grass mixed stand having lower CN ratio compared to the grass monoculture stands. Okay, and this is the effect of drought stress on shoot nitrogen content. This axis shows the total shoot nitrogen content under different moisture treatment. And what we found under moderate and severe drought condition, the total shoot nitrogen content of the red clover, even under mixed stand, is like really lower compared to the well-watered plants. And we didn't see any significant differences in terms of like total shoot nitrogen content in grass, but you can see the green bars like always like sitting at the bottom. That means like total shoot nitrogen content. So this is a gram of nitrogen per plant. So it was lower compared to the grass mixed stand. Same trend during the recovery phase. Again, you can see the total shoot nitrogen content, it was lower under severe and moderate drought stress in red clover compared to the well-watered plants. And again, like green bars, sitting at the bottom compared to the orange color bars. So showing like shoot total nitrogen content, again, it was lower in grass monoculture compared to the grass that were grown with like legumes mixed stand. Okay, then in this study also we calculated the nitrogen fixation in terms of like percentage nitrogen derived from atmosphere. So we similar to previous experiment, we label these plants with isotope, then measure the nitrogen fixation. And similarly, we found severe drought stress reduce the nitrogen fixation capacity compared to the well-watered plants as well as moderate drought stress plants. And so this is drought phase, and this should be the recovery phase. Actually, this is like drought phase, and this is the recovery phase. So recovery phase, again, we found like slight reduction in nitrogen fixation capacity under severe drought stress compared to the moderate and the well-watered condition. Then we calculated, so the title, this title should be changed. Actually, this is the drought phase and this is the recovery phase. We calculated total nitrogen fix. So basically by multiplying percentage NDFA, nitrogen derived from atmosphere, by total nitrogen percent in the shoot materials. So what we found, like total nitrogen fixation, the amount of total nitrogen fix, it was really, really low under severe and moderate drought stress compared to the well-watered plants. So this is drought phase. And also like during the recovery phase, so we found the moderate and severe droughts significantly reduce the amount of total nitrogen fixed in the shoot biomass compared to the well-watered plants. Okay, so so based on these observations, we came to like following conclusion. So drought reduced plant biomass, nitrogen fixation, shoot total nitrogen content in red clover and timothy, and mixed cultures of red clover and timothy has like positive effect on biomass production and total shoot nitrogen content even under moderate and severe drought stress condition. So adding, having this mixed stand is more favorable under drought condition. So incorporating legumes with grass increase the overall forage quality and enhance resilience in drought stress condition. So I didn't present one figure. So we measured the water use efficiency. So what we found under mixed stand, the water use efficiency, it's higher compared with growing grass alone. Okay, so with that, so I would like to acknowledge some people. So the first experiment I presented, it was conducted by one of my master's student, Daniel Todolito. Then the second trial I presented, it was conducted by one of my PhD student, Chaturanga de Silva. and Dr. Ria Lumek took help with like all the microbiome work. And also, like, I would like to thank Dr. Harry Powdell from Agriculture and Agri-Food Canada, Lethbridge, for helping with all the isotope analysis, especially for nitrogen fixation measurement. And also, I would like to thank funding from NSERC, and also support from the University of Alberta and all the people from my lab. And yeah, it's like picture of my lab. And so I'm happy to take any question. At the same time, here is my email address if you want to reach out to me. And I'm also in Twitters and usually check regularly my Twitter account. And yeah, and thank you very much for joining, and happy to take any questions.
Thank you, Malinda. Any questions from anybody? I have a question, Malinda.
Yeah.
I think I believe that we have a number of producers listening to you today.
Mm-hmm.
So, what would you think would be a summary for the producers, those who are growing plants under different environmental conditions?
So we were trying to, do this experiment, like, as a proof of context. And I would like to do this kind of like field experiment, like somehow, in these drought under field conditions, and see, what exactly going to happen. So I'm pretty sure, we might see, same trends under field conditions. I think one of the main thing I want to highlight here, like, I think it's I'm pretty sure, most of the producers, they tends to grow, legumes and grass together. So I think legume plays very important role. And sometimes, what happened, like, the producers have doubt, whether I actually, I really need to inoculate my seeds, or whether I need to, like, use the inoculate seed. I think that is very important, like, to buy seeds, with a reliable source, because, make sure, like, having good nodulation and nitrogen fixation. So what happened in the field, there are some other rhizobia, nitrogen fixing bacteria, right? That can invade the plants and produce nodules, but they might not be efficient as some of the rhizobia, that we inoculate. So we need to make sure, having good nodulation. And I think having the good balance between the grass and legumes is very important in terms of, like, forage quality and everything. And based on our experience, what we found, having like this good balance of like forage and legume stand also help to mitigate some of the drought stress. Definitely they're going to have like less yield, but at least, they can have, like relatively low impact, when we have this grass legume mixed in. But maybe we need to test that, what is the best ratio, in terms of like drought stress. And one thing, that especially drew my attention is this soil nitrogen availability after drought stress. I think this is like like uh farmers, usually grow like, annual crops. They also know, right? When there's a drought stress, the next spring the soil nitrogen available nitrogen is going to be much higher, so they need to adjust their nitrogen application. So forage sector, it's a perennial system, so that needs to be, countered if producers going to apply. I don't have, much background, how producers actually apply fertilizer and everything. So that's maybe that needs to be considered after drought events. So there will be like a lot of nitrogen available in the soil. Yeah.
Were your pots fertilized before you
No,
planted?
we didn't apply any nitrogen fertilizer. We only applied like phosphorus and potassium and other micronutrients. So we didn't add any nitrogen fertilizer. So all the nitrogen came from nitrogen fixation.
That's why the grasses were all yellow, because there
Yes,
was
that's
no
right. Yeah, yeah.
no
Otherwise, it should be.
leaking.
Yeah, maybe we should have included another control like
Yeah.
supplying nitrogen. Yeah.
Any other questions from anybody?
There's questions in the chat.
Okay.
Okay.
So the first questions, the grasses seems to be quite constant in each moisture level. So that's right. I think maybe one of the reason for this observation, I believe, uh, so soil didn't have much nitrogen to support grass growth, because
Yeah.
grass demand a lot of nitrogen. So maybe if we supplied nitrogen, we might have seen kind of like different scenario. So I think maybe plus kind of like reach kind of like optimal levels. I think that may be one of the reason we didn't see any differences in terms of biomass. The second question is, would you expect similar trends in an established stand as opposed to a newly seeded stand? Yes, I think, yeah, I would expect, different stand, because what happened, so once these legumes especially get kind of older, like a couple of years older, some of the nodules can start like senescing, and more nitrogen can be released once they decompose. I expect like more nitrogen release, like newly seeded stand, they're kind of like starting the nodulation, and there may not be like much nitrogen fixation happening, especially during the establishment year. But when it's got like second year and third year, I did a field trials when for my PhD. It was like in Nova Scotia. So we did study using red clover and bluegrass stand. So what we found, so we did multiple years. So what we found, like nitrogen fixation and also available nitrogen in the soil actually becoming, increased, once we progress, like second year and third year. So we can expect like more nitrogen fixation, as well as like more nitrogen release from these legume plants once they get mature. Okay. Also, could you speculate on effect of drought that lasts months or years? Um, that's a good question. I think I because whatever the experiment we conducted in the first experiment, I think we had drought only 3 weeks, and second experiment we had drought only 4 weeks, and I think that's not sufficient to like see, much significant impact. I think if we have like drought maybe like couple of months, or like, even like more period, I think whatever the parameters we measured, we might see like huge effect, especially like, if I talk about like forage production, that's going to be, drastically reduced. And like nitrogen availability, that can like significantly change. Even like soil microbiome, that can like completely change because of this prolonged drought condition. And some of the microbes maybe can significantly reduce, and some of the microbes can change, and their contribution in terms of like nitrogen cycling, carbon cycling, like decomposition, that can really change once plants exposed to like very prolonged drought stress. Yeah, I'm really interested to see what happened, especially after this winter, like how that's going to affect like all the cropping system. Now people talk about what's going to happen, like pest and diseases. Like I saw like some articles and some webinars, people are talking about that can be it can be like the bizarre this year, because it's kind of like mild winter, so we might have pathogens. So similarly, so we might see some significant events yeah, following this winter. um Okay, the next one is, will soil nitrogen availability affect drought mean compensatory plant growth after drought? Yes, that's what we observed. So this available nitrogen can be absorbed by plants, unless we get like heavy rainfall. But usually during prairies, I don't think like we get like much rainfall. Uh, so most of the nitrogen will be available. So plants can absorb most of this nitrogen, yeah, following drought spells. Yeah. And one thing maybe we can measure, like, yeah, in the field, we can actually check installing some light meters. We can see these nitrogen can reach out from the system as well. Yeah.
One other question, if I may ask, Malinda. When I looked at your graphs, most of the cases, I noticed that the error bar was much larger than in the very severe drought compared to the other. Would you
Yeah.
agree with that? Do you think there
Uh,
was a lot more variation in the droughty observation compared to the rest?
uh, Yes, I mean, in this situation, like we didn't want to like remove like any outliers or anything. We just want to like
Yeah,
show like what exactly happened.
yeah.
Most of the people they try to remove outliers and
Mhm.
show, okay,
Mhm.
this is the mean value. But I think there's like much like lot of variability.
Mhm.
But still, I think we still see the changes. So maybe like when we supply enough moisture, plants have kind of like optimal growth. But when it's come to
Yeah.
like drought, maybe the variability is much higher, because we had to provide this drought by measuring, like we had to measure the pot weight every other day. Then we had to
Mhm.
adjust. So that means like we couldn't like precisely, like we say like 20%, we can't like precisely maintain 20%. That can range from like 20%, and sometimes it can even go to like 10% based on the evaporation and transpiration. So
Yeah.
maybe that also need to be considered. Yeah. There's another question. What if efficiency was higher on mixed, and if so, what do you attribute that to? Yeah, I think that's what we found. So we also measured carbon isotope. Based on carbon isotope, we can use it as a proxy. It's called CID, carbon isotope discrimination, and we found water use efficiency was, I think, better on the mixed end. So if so, what do you attribute that to? So maybe what I can say, maybe under mixed and probably plants tends to utilize moisture effectively, maybe compared to like when you grow like grass alone. So maybe it's a good thing. Yeah.
Hey, any other question?
Yeah, I really
Yes.
hope like we can do some more like field trials in the
Yeah.
future.
Yeah.
So recently I'm involved in one of the big grants we received from Genome Canada. So what we are going to do like in this trial, so we are going to have um, rotational grazing and continuous grazing. Then we are going to measure, like we are going to do it in like a Matthias range and Kinsella, and also using some living labs. So we are going to test, how rotational grazing and continuous grazing is going to affect like plant productivity, like soil microbes, soil carbon sequestration, greenhouse gas emissions, everything. So
Mhm.
that's a big project, and we are going to start this year. And I'm pretty sure we get like a lot of new information out of that grant. So it's a like $6 million dollar funded project. So I think we'll have like a lot of information, especially, I mean, this is for like a grazing system. Yeah.
Mhm. Oh,
Yeah, I also like, I mean, Surya, I mean, like to hear like from producer, if they have any, some question, if I can, hear this question. Okay, we have these kind of question, then we can, I like to hear, what are the main problems they have. Then we can, develop, do some experiment and, come up with some answers for some of the questions.
yeah,
Yeah.
with through AFIN, I think that's what we are trying to encourage the producers to have interaction with the
Mhm.
researchers. It's not easy to have good interaction among researchers and producers all the time. And I think we don't have a major conference
Mhm.
where we can actually facilitate that, and that is one of our major goals in AFIN strategic plan. That's what we are trying to do. We're trying to do a major conference almost every year or every other years, where we can bring the researchers and producers together so that they can have real good dialogue and understand each other's problems, understand each other, what they can do, how they can help each other. And I think we are spending money, but at the same time, if some of the research is not done, what the producers are looking for, then it takes long time for anybody to adopt it. So that's the main idea behind our strategic plan. We are trying to bring together the researchers and producers. Let's hope we can do it. We can do it in an effective way, and we would definitely encourage researchers to interact as much as possible with the producers through these webinars, seminars, and also conferences.
Yeah.
There's one more question in the chat.
Yeah.
Okay.
So the question is, would it be safe to assume AMF, arbuscular mycorrhizae fungi levels would be very low to non-existent in a pottex run as opposed to field conditions? Hard to say, because we collected soil sample from the field and that should have spores, mycorrhizae spores, and I'm pretty sure I can say, like, probably we had like mycorrhizae, but of course, I think under field condition, I think we can expect more mycorrhizae. Yeah. Yeah. I think that's one of the area maybe like not tapped by, many of the research scientists, especially in forage system. And yeah, I recently got a, funding from ARDA, that's to evaluate the mycorrhizae colonization in wheat. So maybe that's some some areas probably I will explore in the future. Yeah.
Yeah, if you can find some time, do some specific research on forages so that we can have some of these inoculants available for our forage crops. We don't have, for minor forages, we don't have inoculants available. We would encourage you to spend some time with it, if not full time, but actually develop something that can help our producers. We would very much appreciate that.
Yeah. Yeah, I was
Any
also
other
having
questions?
hard time. I was also having hard time like finding some inoculants for some of my experiment.
Exactly, exactly. I know that. I
Yeah.
was, until 2 years ago, I was very much involved in these things. So now I've retired, but I still have interest in coming up with some of these inoculants that are not available for
Yeah.
sisal milk, vetch, and sandfoin and crops like that. Alfalfa, we can find inoculants, but not for the other two.
Yeah. Yeah.
So if there are no other questions, I think I would thank Malinda for being the first batter in this year's seminar series or webinar series. I think we thank you for agreeing to do this for us, Malinda, and hope that you would keep on giving us good presentations and your experience in your research, and talk to our producers and also learn from producers what they are looking for in future time. We thank you very much for taking the time and spending the time for developing this webinar and presenting it to us. Thank you again from AFIN side. We thank you very much. And one other announcement I should make here, that on 14th of March, we will have our AGM, annual general meeting. We would like people to attend that because we are trying to bring some speakers, those would talk about their life experience with forage crops. And I think you can learn few things from how to grow it, how to market it, and things like that, so that you would benefit from the speakers' experiences. So please come and attend. We have a very diverse speaker group. I think those who would participate in this AGM, it's only one day, and it does not cost very much. It would be at Luduk on 14th of March. Anything else, Amanda, you want to
That's
mention
perfect.
here?
Thank you, Surya.
Okay. No, anything? Anybody else wants to say anything? Parting remarks?
We will have a copy of this video on our website as well. We'll be posting it to the podcast channel. So if you wanted to listen to it again or watch it or share it with somebody else, we'll have that up in the albertaforage.ca website very, very quickly.
And thank you again, Malinda, and good luck with your research. I think I see that you were quite involved already with many projects. And I think that's the right way to start.
Yeah.
You're young, energetic, go ahead and do it. Do it so that after 30 years, you can talk like me. Okay.
All right. Thank you very
Okay.
much, Surya. Thanks for the opportunity, and thanks everyone. Yeah.
Thank
have a
you,
good night.
everybody.
Take
Good
care.
night.
Bye.
Thank
Thanks.
you.
Thanks.
The Sustainable Forages webinar and podcast is a series of informative and engaging conversations about sustainable forage production and management practices. Each episode features experts and innovators in the field discussing a range of topics, including soil health, grazing management, and the latest research on forage crops. Whether you're a farmer, rancher, or just interested in sustainable agriculture, this series is a valuable resource for anyone looking to learn more about the importance of forages in a sustainable food system.